What's going on, besties? Today we're going to be talking about the TEAS version 7 portion of the science exam, more specifically life and physical sciences, and we're going to be covering macromolecules and microorganisms in disease. Let's get started. So let's take a moment to consider our favorite food. While we all have our unique preferences, food fundamentally serves as a source of critical, large molecules necessary for life, known as biomolecules or macromolecules. These can include things like carbohydrates, lipids, proteins, and nucleic acids, each found in varying quantities across different foods. For instance, my favorite peanut butter is rich in both proteins as well as lipids. Although this video is going to reference examples of food containing biomolecules, our focus isn't going to be on nutrition but understanding the major classes of those biomolecules and their critical roles in cellular structure and function. After all, these cells make up the organisms themselves. Before we deep dive into each one of these 4 different types of biomolecules, we must introduce an essential term, the monomer. The monomer acts as a building block. The smaller units composing any large molecule is termed monomers. As we explore each class of biomolecules, we'll discuss their respective monomers if they apply. Before we move on, let's take a moment to appreciate the stunning structure of biomolecules. As we review each one of these, let's think about how we might remember the primary and most common elements that they contain. There's a handy mnemonic called CHO, CHO, CHON, and CHUMP, where instead of an M, we actually use an N. This helps us actually recall the elements in each one of these biomolecules. Carbon is our C, hydrogen is our H, oxygen is our O, nitrogen is our N, and phosphorus is our P. So you may question, does this imply these are the only elements that we're going to find in these biomolecules? Certainly not. However, understanding the common elemental composition and their arrangements within these biomolecules is crucial because their structural configurations significantly influence their functions. Let's begin by exploring the class of biomolecules known as carbohydrates. Common sources like bread, pasta, fruits, and vegetables are all typically very rich in carbohydrates. The basic building block or monomer of a carbohydrate is known as a monosaccharide. A prime example of a monosaccharide is glucose. Glucose plays a critical role when it comes to cellular respiration, where it produces ATP, which is often referred to as the cell's energy currency. When 2 monosaccharides link together, they form what is known as a disaccharide. Here's a memory trick for you. You'll notice many sugars share a common suffix, OSE, like glucose, maltose, lactose, sucrose, and fructose, which helps indicate that these are carbohydrates. When many monosaccharides are linked together, they create a polysaccharide, which represents a more complex form of carbohydrate. If you're having any difficulty remembering which saccharide is which, I use the memory trick mono, di, and poly, because mono stands for 1, di stands for 2, and poly stands for many. Carbohydrates plays a crucial role in the structure and function of cells across various organisms. For instance, plant cells have cell walls composed of specific carbohydrates called cellulose. Similarly, fungi have cell walls made of a carbohydrate known as chitin, which interestingly is also the material that forms the exoskeletons of insects. A vital function of carbohydrates such as glucose is its role in energy production. Like we talked about before, glucose is instrumental in generating ATP, the primary energy carrier in cells, making carbohydrates a rapid and accessible energy source. Furthermore, this energy can be stored in the form of polysaccharides, complex carbohydrates like starch in plants and glycogen in animals, which serve as an energy reserve to be utilized when needed. Next, let's discuss lipids, which encompass fats and oils. Common sources include items like butter and olive oil, and they represent a remarkably diverse category of biomolecules, encompassing several types, including triglycerides, phospholipids, and steroids, just to name a few. While many lipids are composed of glycerol and fatty acids, this isn't universally true for all lipids. Lipids also do not have a true monomer because they do not have repeating units like we saw with carbohydrates. However, a common characteristic when it comes to most lipids is their hydrophobic nature, meaning that they tend to repel water. This hydrophobic nature means that lipids generally do not dissolve in water, unlike most other biomolecules, which makes them unique in their interaction with water and their role in the biological systems. Lipids play a crucial role in their structure and function of organisms and their cells. The cell membrane is the fundamental structures in all cells, whether it's plants, animals, or other organisms. This essential barrier is largely composed of phospholipids, arranged in a phospholipid bilayer thanks to their structural properties. Beyond structural roles, lipids are vital in long-term energy storage. While carbohydrates do provide quick energy, lipids store energy for longer periods, which organisms can tap into when carbohydrate sources are depleted. Lipids also play a significant role in insulation. This includes electrical insulation, such as the myelin sheath surrounding many neurons, which enhances the speed of electrical impulse transmission. Thermally, lipids are integral for temperature regulation, evident in the thick layers of blubber like we see in marine animals such as the harp seal, or even in an oversized kitty cat, which helps them retain heat in frigid temperatures. Another significant function of lipids is hormonal activity. Many lipids act as hormones, serving as key chemical messengers that regulate various biological processes throughout the body. Next up, we have proteins. Common protein sources include meat, nuts, beans, and even eggs. Proteins are polymers made up of amino acids, which serve as their monomers. There are various types of amino acids which link together to form proteins. Proteins are crucial for both structure and function of an organism and their cells. Structurally, proteins are major components of tissues such as muscles. They are also found in other body structures such as hair and collagen. Proteins play an essential role in cell membrane integrity by forming protein channels and receptors, which are vital for transporting substances across the cell membrane and for cell signaling, enabling these cells to communicate and coordinate their actions. Functionally, most enzymes are proteins. Enzymes catalyze biochemical reactions in the body, aiding in both the synthesis and breakdown of substances during metabolical processes. Proteins are also fundamental to the immune system. Antibodies produced by immune cells are proteins that play a key role in defending the body against pathogens. Furthermore, several critical hormones, including insulin, are also proteins, highlighting their role in regulating physiological processes. When it comes to genetics, although genes are composed of DNA, many genes encode for proteins, underlying the genetic basis for building and regulating the body's proteins. This brings us to our final category, nucleic acids, which encompass both DNA and RNA. We'll explore nucleic acids in greater detail when we talk about genetics. The monomer of a nucleic acid is known as a nucleotide, in terms that conveniently resemble nucleic acids, making it much easier to remember. Nucleotide, nucleic acids. Nucleic acids are essential across all life forms, whether it's plants, animals, fungi, or even bacteria, as they contain the genetic instructions necessary for introducing cellular activities. In terms of their importance inside of your cells, nucleic acids hold all your genetic information in the form of DNA and RNA. This genetic content is crucial for coding your traits that define your structure and your function, influencing everything from physical appearance to psychological processes. So our first practice question: what is the monomer of carbohydrates? Is it A, nucleotide, B, amino acids, C, fatty acid, or D, monosaccharide? And the correct answer is D, monosaccharide. Remember, monosaccharides are the simplest form of carbohydrates, and they serve as the monomers for those more complex carbohydrates. What type of lipid is primarily used by the body for long-term energy storage? Is it A, steroids, B, phospholipids, C, triglycerides, or D, waxes. And the correct answer is C, triglycerides. Remember, triglycerides are the main form of stored energy in your body and are found in a lot of your fat cells. Which type of macromolecule plays a key role in forming cellular membranes? Is it A, nucleic acids, B, carbohydrates, C, lipids, or D, proteins? And the correct answer is C, lipids. Lipids, specifically phospholipids, are fundamental components of cell membranes. Now we're going to discuss infectious agents, often referred to as microorganisms. However, it's crucial to note that not all agents that we're going to cover today qualify as living organisms. Some actually aren't even alive at all. They're neither cells nor considered life forms. So for clarity, we're going to refer to them as infectious agents instead of microorganisms. These agents can enter our bodies, which act as hosts and potentially cause injury, disease, or even death. When discussing anything that can cause disease or injury, we're going to use the term pathogenic. This term can be broken down into two parts: patho, which means disease, and genic, which means causes. Thus, pathogenic refers to an infectious agent capable of inducing disease. Another term that you might encounter when you're taking the TEAS is virulence, which describes the severity or harmful potential of a pathogen. It's important to understand that virulence doesn't necessarily correlate with more damaging outcomes. So for instance, a more virulent strand of a virus may not always cause more severe disease, but could be more adept to invading immune systems, more transmissible, and it could even be more capable of circumventing vaccines, or in the case of bacteria, antibiotics. So in summary, pathogenic means capable of causing disease, while virulent refers to the potential severity or nastiness of a pathogen. So let's start off with our favorites, viruses, familiar to many and very significant in the study of microbiology. Viruses are tiny entities ranging from anywhere between 20 to 400 nanometers in size. Notably, viruses are not cells and they're not actually considered alive. They remain motionless on surfaces like benchtops or the ground outside of a host organism. To be active, a virus has to enter a host, whether it's an animal or it's a plant, and once it's inside, it behaves as an intracellular obligate parasite. This means that it's compelled to reside within a host cell to function, specifically to replicate and perform activities. Viruses are classified based on structural components as well as genetic material. The outside shell of a virus is known as a capsid, and the genetic core inside is referred to as a genome. Thus, viruses can be categorized by their capsid structure and whether their genome consists of DNA or RNA. Additionally, some viruses have an envelope, which is derived from the host cell's membrane as the virus exits the cell, enveloping itself for protection and aiding in infection. The impact of a cell on the body and its pathogenicity depends on where it infects. Different viruses target specific regions, such as the respiratory tract, your gastrointestinal tract, systemic, skin, central nervous system, just to name a few. For instance, the common cold, influenza, and COVID affect the respiratory system, while rotavirus and mumps is prevalent in your GI tract, especially in children. Systemic infections like chickenpox and HIV can affect the entire body, and skin-specific viruses like we see with HPV can cause cervical warts, or even worse, cervical cancer, due to that cellular mutation induced by viral genetic material integration. Viruses attach to a host cell via structures like spike proteins on their capsid, which bind to specific receptors on the cell membrane. Upon attachment, the virus injects its genetic material into the host cell, hijacking the cell's machinery to replicate new viruses. This is often going to lead to the host cell's death, contributing to the disease. In some cases, like we see with HPV, that integration and mutation of viral DNA can lead to cancer. Fortunately, though, for many of these viruses, vaccines are available, providing protection by priming the immune system against these infectious agents. Moving on, let's talk about bacteria, which marks a significant jump in scale. Bacteria can range anywhere from 0.2 to 15 microns in size. To put this into perspective, 0.2 microns is equivalent to 200 nanometers, and 15 microns is equivalent to 15,000 nanometers. From here until the rest of our examples of microorganisms, unlike viruses, we're dealing with living organisms, cells that are very much alive. Bacteria are classified as prokaryote, meaning that they have DNA, but it's not contained within a membrane-bound nucleus. Instead, it resides in an area called a nucleoid. Unlike eukaryotic cells, most bacterial organelles are not enclosed by membranes. A key structural feature of bacteria is their cell membrane. It's similar to that of other cells, but more permeable, which means that they are more susceptible to swelling and bursting under fluid pressure. Fortunately, bacteria have a protective cell wall, which helps maintain its structural integrity against such pressures. This cell wall is crucial for the bacteria's survival and is composed of a network of proteins and sugars. Bacteria can be further categorized based on the composition of these cell walls into two types, gram-positive and gram-negative. Our gram-positive bacteria have a thick cell wall, while gram-negative bacteria has a thinner cell wall but an additional outer membrane. Additionally, bacteria can be categorized by their shape. Cocci is more spherical and bacilli is more of a rod shape. The organization of these shapes can be indicative. For example, cocci that group in a grape-like structure can be known as staphylococci, and those that form in long chains are known as streptococci. One disease example could be chlamydia, which is a bacteria that requires intracellular environments to access energy and carry out its functions effectively. As a result, they can also lead to cellular damage, ultimately causing scarring. Such scarring can occur in the reproductive tract, especially in females, potentially leading to infertility. In the eyes, this scarring can actually impair vision and lead to various types of visual dysfunctions. Another example would be mycoplasma, notably one of the primary causes of community-acquired pneumonia. Mycoplasma is unique because it lacks a typical bacterial cell wall, which allows it to live both inside and outside of host cells. We also have bacteria like Streptococcus, which can cause conditions such as strep throat, primarily affecting the respiratory tract. Staphylococcus aureus, aureus meaning golden, which is a common cause of skin infections. Additionally, there is Clostridium tetani, which is particularly interesting because it produces exotoxins. Gram-positive bacteria like Clostridium tetani release exotoxins that can cause severe damage by infecting motor neurons with skin wounds, leading to tetanus. This condition is life-threatening and it can cause muscles to stiffen uncontrollably. In contrast, gram-negative bacteria produce endotoxins, which are part of their outer membrane. When these bacteria die and break down, they release those endotoxins, triggering intense inflammation as well as other serious health issues to the host. As we progress to larger cellular structures, we encounter fungi, which range in size from 2 to 200 microns. Fungi, like the cells we find within our body, are eukaryotes, meaning they possess membrane-bound organelles, including nuclei. Unlike bacteria, fungi have cell walls made up of chitin, which is a polysaccharide also found in the exoskeletons of insects like spiders. Fungi has the capability to reproduce both sexually and asexually, often relying on spores, which function similarly to seeds to spread and propagate. When fungi causes diseases, the condition is referred to as mycosis, which can manifest as superficial or deep infections. Superficial fungal infections primarily affect the skin, typically arising from fungi that releases enzymes irritating the skin and causing symptoms like itching and redness. A common example is tinea, which is known as ringworm, thought to not be actually a worm but a fungal infection. Depending on the affected area, tinea may be called tinea capitis if it's on the scalp, or tinea pedis if it's on the foot like we see with athlete's foot. Fungal infections can also impact mucous membranes, leading to a condition known as thrush, which can occur in our mouth. We have oral thrush, or it can occur into the genital area known as vaginal thrush. More severe and deep fungal infections may involve internal organs, like the lungs or bloodstream, particularly in immunocompromised individuals. In such cases, fungal infections can lead to serious health complications and are a common cause of mortality in people with weakened immune systems, such as those with HIV. Protozoa, also referred to as protists, range in size from 1 to 50 microns. These organisms are eukaryotic and usually unicellular like we see with fungi, or they're typically multicellular. Protozoa possess all the organelles necessary for metabolic functions, similar to those found in our cells, including nuclei and mitochondria. Examples of diseases caused by protozoa include malaria and giardiasis. Malaria begins with the protozoan's life cycle inside the mosquito's gut and saliva. When a mosquito bites, it injects what can be similar to cysts into the bloodstream, which then travels down into our liver. Here, they mature within the hepatocytes before bursting out and infecting red blood cells. Within these cells, the protozoa reproduces asexually, leading to the typical symptoms we see with malaria. Once released from the red blood cells, the protozoa enter a gamete form, ready to be picked up by another mosquito, continuing that transmission cycle. It's important to note that the mosquito only serves as a vector. The actual infectious agent is the protozoa. Giardiasis, on the other hand, is a digestive disease which enters the body through contaminated food and water. The cysts are ingested and travel to the upper digestive tract, where they hatch and multiply through mitosis. They then attach to the digestive tract walls, absorbing nutrients and causing irritation and inflammation. This process often results in symptoms like severe, watery, and sometimes bloody diarrhea. Eventually, the protozoa produces more cysts, which are excreted in feces and often contaminate food and water sources, perpetuating the cycle of infection. Finally, we move on to animals that can be classified as helminths, which are worms or ectoparasites. Helminths are multicellular eukaryotic organisms that range significantly in size, from as small as 3 milliliters all the way up to as large as 10 meters. In terms of classification, helminths can be divided based on their shape. They can be roundworms, flatworms, or flukes. Roundworms, such as hookworms, typically inhabit the gastrointestinal tract and have a cross-section circular appearance. Flatworms, like tapeworms, are often very long and have a ribbon-like shape. Flukes, such as liver flukes, are typically leaf-shaped. As for ectoparasites, these include organisms like mites, fleas, and bedbugs. These parasites are primarily known for causing skin irritation and inflammation. They attach to the skin, feeding off the host, and often leading to discomfort and allergic reactions. If we consider creatures like ticks, they can transmit bacteria through their bites, known as Lyme disease. A tick bite introducing bacteria into the skin can potentially lead to significant health issues. Which of the following is characteristic of fungi? Is it A, they are photosynthetic, B, they reproduce by binary fission, C, they have a cell wall made of chitin, or D, they are typically motile? And the correct answer is C, they have a cell wall made of chitin. Remember, fungi have that cell wall, and it's going to distinguish them from plants who have cell walls made of cellulose. Which type of microorganism causes malaria? Is it virus, bacteria, protozoa, or fungi? And the correct answer is C, protozoa. Malaria is caused by protozoa, which is transmitted to humans from a bite of an infected female mosquito. What is a unique feature of viruses compared to other microorganisms? Is it A, they contain both DNA and RNA? B, they require a host cell to replicate? C, they have a rigid cell wall? Or D, they can undergo photosynthesis? The correct answer is B. They require a host cell to replicate. Remember, viruses are unique in that they cannot replicate on their own. They have to invade a host cell and use its machinery to produce new virus particles. Infectious and non-infectious diseases represent two broad categories of illnesses, each with distinct causes and mechanisms. Infectious diseases are caused by pathogenic microorganisms, such as bacteria, viruses, parasites, or fungi. And these diseases can spread directly or indirectly from one person to another. For example, COVID-19 primarily spreads through respiratory droplets when an infected person either coughs, sneezes, or talks. It can also spread by touching surfaces contaminated with the virus, and then touching your face, although it is less common. Similarly, influenza is caused by various strands of the flu viruses and is highly contagious, spreading through respiratory droplets. Chickenpox is another highly contagious disease, most commonly affecting children. This virus spreads through direct contact with a rash or through the air by respiratory droplets when that infected person coughs or sneezes. Non-infectious diseases, also known as non-communicable diseases, are not caused by infectious agents and cannot be transmitted between people or other organisms. These diseases are often the result of genetic, environmental, or lifestyle factors. Examples can include diabetes, which results from the body's inability to produce or effectively use insulin. Heart disease is often caused by a combination of dietary, genetic, and lifestyle factors leading to the buildup of plaque in our coronary arteries. Cancer can have various causes as well, including genetic predisposition, environmental exposures, and lifestyle choices such as smoking and diet. Understanding the distinctions between these two types of diseases is crucial for effective prevention, treatment, and management strategies. Next, we're going to examine the various ways diseases are transmitted and often referred to as modes of transmission. The first category we are going to discuss is going to be direct transmission. This occurs when infectious agents are immediately transferred from a source or reservoir, such as an infectious person, directly into a susceptible new host. Direct transmission can occur in two primary ways, either through direct contact or droplet spread. Direct contact is going to involve physical interaction with bodily fluids from an infected individual, which might include oral secretion, blood, or other fluids like we see with skin lesions, semen, or vaginal secretions. This type of transmission can result from activities like touching, biting, kissing, or sexual intercourse. Droplet spread, another form of direct transmission, requires droplets from respiratory droplets containing those infectious agents. This typically happens when an infected person coughs or sneezes, projecting droplets directly onto someone nearby. Diseases like pertussis, also known as the whooping cough, is often spread this way. For droplet transmission to occur, close proximity is necessary, generally less than 2 meters, such as sharing a small space like a car or a room. It's important to understand that both direct contact and droplet spread are considered direct transmission methods. Next, we'll discuss indirect transmission, which includes several mechanisms, one of which is airborne transmission. Airborne transmission involves infectious agents that are carried on dust particles or within droplet nuclei that remain suspended in the air. Due to their small size and light weight, these droplet nuclei can linger in the air for extended periods of time, allowing them to be inhaled into a host's respiratory system. With airborne transmission, neither direct contact nor close proximity is necessary for the disease to spread. The droplet nuclei can travel and disperse widely across an environment and can infect a host without any direct interaction with the person who was infected. Indirect transmission involves objects and items, often referred to as fomite transmissions. They occur when infectious agents are transferred from an infected person to an object or a surface. These objects, like door handles, tabletops, utensils, or electrical devices, can then harbor the infectious agents for varying lengths of time. When another individual touches these contaminated surface and then touches their face, eyes, nose, or mouth, they can inadvertently introduce that pathogen to their body, leading to an infection. The risk of fomite transmissions depends on several factors, including the type of pathogen, the environment, and the material of the object. For example, hard surfaces like plastic, stainless steel can retain some viruses for days, whereas more porous materials like fabric are less conducive to long-term survival of viruses. Regular cleaning and disinfection of frequently touched surfaces are effective strategies to reduce the risk of disease spread through fomites. Hand hygiene is also crucial in preventing the acquisition and spread of infections through indirect contact with contaminated objects. And lastly, we have vector-borne diseases. They are transmitted when an infected person passes a pathogen to a vector, typically an insect, which then spreads the infection to another person. Without this vector, the disease cannot pass directly between individuals. Due to their complex transmission dynamics, these diseases are among the most challenging to prevent and control. For example, preventive strategies in the fight against malaria, which is spread by mosquitoes, includes vaccinations and environmental tactics. These environmental methods often involve using mosquito nets or removing standing water to disrupt the mosquitoes' breeding grounds. Finally, let's talk about microscopes. To start, it's essential to grasp a few key terms when understanding how microscopes work. Magnification is the primary concept often associated with microscopes, highlighting their ability to enlarge small objects into observable sizes. Another critical term you're going to need to know is resolution, and that's the ability of a microscope to distinguish between two closely positioned objects as separate entities. Without adequate resolution, two proximate objects might appear as a single blurred entity. For instance, a microscope with a resolution of 0.2 microns requires objects to be at least 0.2 microns apart in order for them to be distinct from one another. Let's explore various types of microscopes, each designed with both magnification and resolution in mind to effectively visualize and study specimens. First up, we have the light microscope, which is the type of microscope you typically see in biology classroom settings. As the name suggests, these microscopes utilize light to visualize specimens. The most common type of light microscope is the bright field light microscope, which typically displays a darker image against a light background. Alternatively, there are dark field light microscopes, which includes a component known as a light stop. This element blocks most of the direct light that we see through the light source. And because of this, the only light that reaches your eyes is the light that has been reflected or refracted by structures within the specimen themselves. This technique creates a visual effect where lighter images appear against a dark background, enhancing the contrast and detail of the specimen. Light microscopes utilize light to visualize specimens, but not all microscopes are going to operate by this principle. For instance, to observe extremely small entities like viruses, which are typically smaller than your average bacterium, more advanced technology is necessary to enhance magnification and resolution. This is where electron microscopes are going to come into play. Electron microscopes use beams of electrons instead of light to create images of the specimens. Among electron microscopes, we have two. We have transmission electron microscopes, known as TEM, which are particularly effective in examining the internal structures of a specimen. And on the other hand, we have scanning electron microscopes, known as SEM, which are better suited for detailed visualization of three-dimensional structures of a specimen. Let's finish off with some practice questions. Which microscope would be best for viewing the detailed external morphology of a microorganism? Is it A, light microscope, B, transmission electron microscope, C, scanning electron microscope, or D, fluorescence microscope? And the correct answer is C, scanning electron microscope. Remember, SEMs are ideal for examining surface structures in high detail as they scan the surface with a focused beam of electrons, providing us with a three-dimensional image. What type of transmission involves infectious particles suspended in the air over time and distance? Is it A, direct contact, B, droplet transmission, C, airborne transmission, or D, fomite transmission? And the correct answer is C, airborne transmission. Remember, our airborne transmission refers to the spread of infectious agents through droplet nuclei that are going to remain infectious and suspended in the air over a distance and time beyond 1 meter. Which disease is known to be transmitted through droplet spread? Is it A, HIV, B, hepatitis C, measles, or D, influenza? And the correct answer is D, influenza. Influenza is well known to be transmitted through droplets that are expelled from an infected person either coughing or sneezing, requiring close proximity to spread. I hope that this video was helpful in understanding everything that you're going to need to know when it comes to macromolecules, microorganisms and disease, the spread of infections, as well as microscopes. If you have any additional questions, make sure that you leave them down below. I love answering your questions. Head over to nursecheungstore.com where there is a ton of additional resources to help you ace that TEAS exam the first time. And as always, I'll catch you in the next video. Bye!